Good morning, everyone, and welcome back to the Share Series Monday Management Update. My name is Remy Bernarda, and I'm a partner with Investor Relations Advisory Solutions. I'm excited to introduce you to Alterity Therapeutics, a company that we believe is a compelling investment story as they are looking to make a meaningful difference in the lives of patients with a rare disease. Before we get started, I wanted to remind the audience to use the question button on the top of the video player to submit any inquiries directly to us during the session. With that, let's get started. I would now like to introduce Dr. David Stamler, who is the CEO of Alterity Therapeutics. David, can you please provide a brief overview of Alterity, as well as a short background on yourself and your team and your history at the company?
Thanks, Remy. Sure. Alterity is a global company with our U.S. offices based in San Francisco and our international office based in Melbourne. We have a long history of developing drugs for neurodegenerative diseases, and I joined the company in 2017, originally as the Chief Medical Officer. And then about 2.5 years ago, took over as the Chief Executive Officer. I am very happy that shortly after I joined the company in 2017, key members from a former company that had worked with me joined me, and I'm proud that as a group, we had previously brought drugs to the market in the neurology area. So with this focus, we've been working on advancing our lead molecule into the clinic for a rare disease that I'm sure we'll talk about in a minute.
Great. So you mentioned that Alterity is focused on neurodegenerative diseases. Can you talk a bit more about the diseases you are targeting?
Yeah, so we're focused on Parkinsonian disorders, and Parkinsonian disorders generally include neurodegenerative diseases that feature motor symptoms typical for Parkinson's disease, and that's where it gets its name, Parkinsonian from Parkinson's disease. So as you can imagine, Parkinson's disease is the most common Parkinsonian disorder, but there are other disorders that fall into this class, and one of these is our first indication, or the first disease that we're treating. And it's referred to as, or called multiple system atrophy, which I know is a mouthful for the non-scientists in the group. So I'll just refer to it as MSA. And that term, multiple system atrophy, really just implies that there are multiple regions of the brain that are affected with this pathology. Now, just a word about MSA as a disease. It is a rare disease. There's perhaps 15 to 20,000 patients with MSA in the United States. And as I mentioned, it features the motor symptoms that are typical of Parkinson's disease. So the shuffling gait, the slow movement, the tremor that many of us have seen in friends or family members, or even older individuals on the street. But in addition to those motor symptoms, the patients with MSA have involvement of other systems that affect blood pressure. So patients have a difficulty maintaining their blood pressure when they stand up or have a large meal. They have difficulty with bowel or bladder function. They have impaired sweating. So these are all things that have a demonstrable effect on their quality of life. The other thing that's, I think, noteworthy about MSA compared to Parkinson's disease, although both diseases have similar underlying pathology, what's notable is that MSA progresses about 3 times as rapidly as Parkinson's disease does. So while a typical Parkinson's patient will live perhaps 20 or 25 years after they're diagnosed, a typical MSA patient will only live 7 to 8 years. So it's a multifactorial disease affecting many systems, but it also is a very aggressive disease. And that's why we're really focused on trying to find a new therapy that really slows the course of that disease.
That's great. So I wanted to talk a little bit about how you are addressing these diseases. And just for the audience, we do have a couple of slides for, to walk through just to sort of help emphasize and clarify some of these discussion points. So we'll pull up that first slide. And, David, can you summarize how you are addressing these diseases you talked about?
Yeah, so our aim is to really rescue neurons, and you see that on the far side of the slide. And we do that by binding and redistributing the excess amount of iron that is increased in areas of pathology. Now, if you ask a neurologist who treats Parkinson's disease or MSA, what do you see if you scan their brain? They'll tell you that there's clear evidence that there's increased iron in the areas of pathology in the brain. Now, what that excess iron does is it actually leads to misfolding of proteins and oxidative stress. So our overall strategy is to reduce the excess iron that's in the brains of these patients, to reduce that protein aggregation and protein misfolding, and that protein is really important for neurons to communicate and reduce the oxidative stress. And then the net effect is to really is to rescue the neurons in those multiple brain regions. So by following this cascade, the overall goal of rescuing neurons is to preserve function in these individuals with the disease. And the net effect will be to improve quality of life. So unlike many of the existing therapies that address the symptoms of these diseases, this is really aimed at modifying the course of the disease and trying to improve the quality of life of individuals.
Which is great. So your lead asset targeting this is taking a very rigorous and scientific approach for development. Can you walk us through a little bit now in more detail about how ATH 434 works mechanistically and why this method makes sense to treat MSA and potentially Parkinson's disease?
Sure. So what you see on the slide is what I was just referring to, which is the increased brain iron in a patient with MSA. And the data on the left side of the slide is from an autopsy study from about 30 years ago. And this is from patients with MSA. And you see the patients in blue and then the healthy controls in green. And the investigators compared the amount of iron in the different brain regions in these patients versus the controls. And you can see where the orange arrows are on the left, that it's pointing to two areas where there's increased iron. And those are notable areas of pathology for patients with MSA. And it's really this iron that we're really trying to reduce. And by trying to reduce that, as I had shown you, we're trying to break that cycle that's leading to the neuron loss. Now, if you fast forward about 25 years from that publication, there's been tremendous advances made in terms of identifying and measuring that iron in the brain of living patients, which is really, obviously our focus. And what you see at the orange arrow on that color MRI corresponds to what you see from the autopsy study. You see that dark red staining material that corresponds to the increased iron. And this is important for two reasons. Number one, this helps us identify the patients that we want to recruit into the clinical trial. That's really important because early on, MSA can look like Parkinson's disease, and you don't want Parkinson's patients coming into a trial of MSA. That could really confound your results. And the second important reason is that this becomes an endpoint for what we want to measure in the clinical trial. We've shown in animal studies that we can reduce the iron in the brains of animals with experimental MSA and Parkinson's disease. So the goal is to really target this iron and measure it in the clinical trial.
Okay, and so as you've gone through development, Alterity's taken a somewhat unique approach. You completed a successful phase 1 trial and then started a natural history study you call BioMuse to understand this MSA population and intended for the phase 2 trial. Can you talk about that BioMuse study and the impact you have seen?
Sure. The BioMuse study is really important because before you start to do a clinical trial, a phase 2 trial where you're testing your drug, it's critical that you understand your patient population. Now, MSA as a disease is very well studied, and it's been characterized at academic centers in Europe and the United States dating back 30, 40 years, and there are a lot of publications. However, those publications tend to look at late stage patients when the investigators, thought that, were confident the patients had the disease. But these patients are really too far advanced to easily demonstrate the effectiveness of a drug. So we wanted to study early stage patients for a variety of reasons. Number one, we thought they had the best function to preserve. We thought we could, offer the best clinical benefit to these patients. So we decided to do this natural history that we refer to as BioMuse. And the objective is stated on the slide quite clearly. We really wanted to look at the clinical measures and the biomarkers in these early stage patients. And I'll tell you a bit more about why we wanted to look at the biomarkers in a moment. The design is an observational study. So even though this is our clinical trial that we're doing with our academic partners at Vanderbilt, and we're really interested in the data, there is no experimental treatment. And this is because what we really want to do is just observe the disease. We want to see what the patients look like with early disease. We want to look at their biomarkers, and we want to see how they progress. And then we want to take this information and then learn from it and roll it into the phase 2 clinical trial. And that's really what we've done. Now, just a word about the biomarkers. Biomarkers are, as the name implies, they're biologic measures, and they are measures of disease, but they're not a measure of disease that the patient can feel or experience. So it might be a blood test, or it might be an abnormal MRI finding that you see on the left side of the slide. And what this does is this allows us to assess how the disease progresses. And this is really a contemporary way of drug development that we're moving towards, that we look at these biomarkers, helps us identify the patients, but it also is something that we can track during the course of a trial. And it can give us an early indication of a drug effect before you actually might see what a patient is experiencing. So now we enrolled about 20 individuals with MSA, and we then followed them for a year. And the goal of this therapy, as I mentioned, was to learn for the phase 2 study, to optimize patient selection, to assess the endpoints with precision and pilot clinical measures. So what you see below optimizing patient selection, like I showed you early on with an advanced patient, this is an early stage patient, and we showed that we can now measure the iron in an early stage patient, and that was an important finding. The next thing we demonstrate is we're able to develop our own template that will enable us to measure the brain iron in individual regions with greater precision. And if you think about this is kind of like a map. And if I showed you a map with like a standard template, what's in this green box would be kind of blurry. You couldn't see the difference between these gray and these different gray areas. And that's really important so that we can measure the increased iron in the individual regions. And then finally, we piloted clinical measures in this trial to see if we thought we could detect relevant changes. So this is a wearable sensor that's typical in trials with Parkinson's disease, and now we've shown it works in MSA as well, where you can see this is an individual patient's decline in motor performance in an outpatient setting. So this is the minutes of time they walk per day averaged over a 2-month period. And you can see that after the first 2 periods, this patient really drops off. And this is important because this helps us measure clinical performance that you can't measure in the clinic. And it's really all these things that we learned from the BioMuse trial that we decided to implement into the phase 2 trial.
And just to clarify on that or reiterate, this is really to help distinguish to ensure that you have the right patient population coming in.
Exactly. So what we did after we finished this trial was we then took the 20 or so patients that were enrolled, and then we looked at their clinical criteria, their biomarkers, and it actually gave us the opportunity to go back and change the protocol before we launched it so that we could have even greater precision. We thought we had the best protocol. We did the BioMuse trial, and then we learned that that we could actually improve on this. And actually, the investigator of the trial said, he was very gratified that we did this trial, that we learned much more about how to select patients for the phase 2 trial. And I think what's a unique aspect of this trial is, although there are a handful of competitors out there doing MSA trials, we believe we're the only sponsor that is using several different biomarkers to actually recruit patients into the trial. And as I said before, it's really vital that we know we're getting the right patients coming into the trial.
Which is great. So let's now talk about that phase 2 trial. If you can walk us through what that trial looks like and the key timelines we should be aware
of. Sure. So what you see on the left side of the slide is a diagram for the phase 2 trial. So just to back up a little bit, the trial is a randomized, double-blind, placebo-controlled trial. So if you will, the gold standard for conducting a clinical trial. We are studying, as mentioned before, early-stage patients who are still ambulatory. They'll be randomly assigned to one of 3 treatments in equal numbers: high dose ATH 434, low dose 434, or placebo. Then they will complete 12 months of treatment, and what they will get at baseline and then throughout the study are MRIs. They're going to have spinal tap that's going to look at various biomarkers, and then other biomarkers in their blood. And then after treatment, we'll look at the clinical measures, and we'll also look at the biomarkers. Now, the primary endpoint of the study, which is really the endpoint that you're most interested in demonstrating drug effect on, is shown on the right side of the slide, and that's change in brain iron on MRI, and that corresponds to what I showed you before on earlier slides. These are actually data from the BioMuse trial, but it gives us confidence that we're looking at the right thing. We and others have shown that if you compare the brain iron, which is on the y-axis, with the disease severity as measured by the Unified MSA Rating Scale on the x-axis, that there's a nice correlation between the two. And this is what we're really hoping to do, to halt or reduce the brain iron in these individuals, and to slow down the course of disease and reduce disease severity.
Okay, that's great. And so I think we're going to have probably about 8 minutes or so for questions. But before we take questions from the audience, is there anything you would like to add to sort of close this out?
Yeah, I mean, first of all, we thank you for participating and following along with the exciting work that we're doing at Alterity. And next, I would say that what we've learned so far from not only the phase 1 and the non-clinical work that we've done, but from the natural history study, gives me a lot of confidence that we are doing a unique development program for really a terrible disease that has no approved treatments. And I'm really confident in our approach, and I'm really happy with the great efforts that our team has made to advance this drug candidate for this rare disease that has no approved treatment. So with that, happy to entertain any questions anyone may have.
Wonderful. So we have a couple questions that come in. The first one is, when you report the data from the phase 2, what should we expect to see? And what would a positive trial look like from your standpoint?
Sure. So when we report the data, we'll be reporting on the primary and the secondary, the so-called secondary endpoints. So we'll be reporting on the change in the brain iron with treatment compared to what happens with placebo. And we'll also comment on the change in clinical parameters, whether it's disease severity or measures of change in blood pressure or change in that walking parameter that I showed you, walking time, or even urinary function, which is another source. So those are what we'll be reporting on. What I would say a positive result will look like is really a reduction in brain iron for active treated patients compared to placebo, as well as some sort of a clinical benefit. We don't know if it's going to be on overall disease severity or measures of blood pressure or bladder function. So it's really reduction in iron and some evidence of a clinical benefit.
Okay, and can we talk about timing a little bit? The next question is, when will the phase 2 trial read out and complete?
Right. So we haven't guided on when we're going to release top line data, but our current guidance is that we will complete enrollment at the end during third quarter, and more like September timeframe. And then with 12 months treatment, that would take us 12 months later, and then we need a few months to lock and to clean the data, do the statistical analysis, et cetera. So that's really the time frame that we're looking at reporting these results.
Okay, great. And one of the other questions just came in was about, what are the next milestones? So that would be the answer to that question. And then overall, what is your current financial position?
Right. So we last reported at the end of first quarter that we had about $21 million Australian in the bank, and that in conjunction with anticipated inflows of cash through other sources, we expect will carry us through the end of the phase 2 study. So we'll be able to report this out.
Okay, great. So what are the next steps after you complete the phase 2 trial?
So after we complete phase 2, then assuming that we have a positive result, the goal then would be to start planning for the next phase, the definitive study. And what we would do is take and analyze all the key data, then arrange a meeting with the FDA, discuss our plans with them, and then launch into what we think would be the definitive clinical trial.
And so that would begin, can you just give a little more clarity in the audience, sort of how that works, like as far as the phase 3 goes, for those that might not be familiar?
Sure. So then just a little bit more clarity. So let's just say from time 0, when we get the top line results, we would then request a meeting with the FDA. We would actually know, and that typically can take anywhere from, depending on the schedule, anywhere from 3 to 6 months to get that meeting. So let's just say the 3-month mark, we would get the meeting with the agency. We would then, based on the results of the phase 2 study, we would learn how big the next study would need to be. We would then design that trial, choose our endpoints, and then put a package together, then go talk to the FDA. So then the goal, and that would be for the so-called definitive phase 3 trial or confirmatory trial, that typically would begin that trial about 6 months after we got the top line results from the phase 2. So in the background, we're doing a lot of other things. We're not just waiting for the results, we're manufacturing. We'd be manufacturing all the drug, we'd be packaging it up and packaging it in different scenarios. So we knew we could cover all potential options that would come out of the phase 2 trial. So that's really the goal. And then after that, we're off off to the races, so that we're off to conducting the definitive phase 3 trial.
And we also got a question of where is the trial running? You've said globally.
Yeah.
What does that mean?
Yeah. So because MSA is a rare disease, early on we decided we need to conduct the trial in multiple regions. So we have centers in the United States, of course, and we have centers in Australia and New Zealand as an Australian headquartered company. But we also have centers in key European countries, and we've disclosed and are enrolling in the UK. We're enrolling in Italy, and there are a couple other European countries that we expect will be coming online very soon. So a rare disease, but recruitment is going nicely. We've identified excellent academic centers that really understand this disease, and that's really important because there's a lot of skill skill required to identify these patients in conjunction with our clinical protocol.
Okay, that's great. So I just got notification that we are out of time for today. So thank you, David, for telling the story. If there's additional follow-up questions, we're happy to answer those offline. Just wanted to say thank you to the audience for everyone and your interest in the Share series. Up next today is Odyssey Marine Exploration. Thank you.